Hello, thank you for your invitation today. My title of my presentation is the psychological trauma related to oxidative stress and inflammatory implications. I will begin my presentation for today referring to the history of trauma research going back 2000 before Christ at the Egyptian Kahun papyri, where we found the first evidence of trauma-related literature with regards to hysteria. And if we jump in the timeline, we found at 1750, Pierre Briget, who is considered to be the father of modern study of the trauma field, who has studied trauma through the hysteria somatization filter. And not until 1980 has the PTSD been introduced in the Diagnostic and Statistical Manual. In here, we can find some definitions of the stress types. Firstly, you can see the minor life stress, which describes daily life hassles. And depending on the severity, the stress definition can extend up to severe life stress, referring to stress able to produce severe strain, like the death of a loved one. A typical paradigm of severe life stress can be referred to the Marie Antoinette's case, who during the French Revolution has been condemned to decapitation, and just the night before her being executed with beheading, her hair turned white, and we now know that this was due to the oxidative damage, the oxidative stress, and the damage of the melatonin of the hair. And keeping on with the definitions, we can come across to the early life stress and the childhood maltreatment, which refers to specific forms of abuse during childhood. And last but not least, we should refer the post-traumatic stress disorder, which is a completely recognized and well-described psychiatric condition, which has been studied throughout studies on Vietnam War veterans. Just a couple of decades ago, there has been a notice that early life stress has been presented with a dose-response effect in relationship with psychiatric and inflammatory disorders in adulthood. Another notice has been that early life stress has been linked with major psychiatric disorders, and nowadays we are discussing about the inflammatory etiopathogenetic substrate of the major psychiatric disorders. Subsequently, a question has been risen with regards to the relationship between early stress, trauma, inflammation, and oxidative stress. And for us to answer this question, we need to delve into the metabolic field, the basic research, and the definition of the metabolism is the interaction of the ingredients of the food constituents with the aim to produce energy, to form structural constituents, and to dispose toxic products. All these metabolic procedures take place in the human cells, and especially in mitochondria, where oxidative phosphorylation is taking place, where apart from basic roles with regards to energy production and toxic products disposal, plays role to vital functions of the cell, like cell signaling and neural differentiation. And here we can have a look at different categories of oxidative molecules like reactive oxygen species, reactive nitrogen species, enzymatic antioxidants, and non-enzymatic antioxidants. And in here you can see the oxidative phosphorylation cascade. And in the red boxes, we can see the enzymatic antioxidants like xanthine oxidase, dismutase, glutathione peroxidase, and catalase. In blue boxes, we can see the non-enzymatic antioxidants like vitamin C and vitamin E. And in these green circles, we can see the free radicals. We now know that the free radicals, if in excess, can cause DNA damage, apoptosis, protein carbonylation, and lipid peroxidation, with the final end product toxic ones like thiobarbituric acid, malondialdehyde, and 8-hydroxyl-deoxyguanosine. And in here we can see how the lipid hydroperoxide can be formed, and the initial stage is the loss of the proton from an unsaturated lipid from the cell membrane, and subsequently there is the propagation phase with the oxidation of the lipid radical, where the lipid peroxide is being formed, and after that, in a second step, the peroxide radical borrows a proton from an adjacent unsaturated lipid to form finally the lipid hydroperoxide. This hydroperoxidation is suspended by the effect of the antioxidants. And all the knowledge in regards to the oxidative stress comes from preclinical and clinical studies. Here we can see animal studies relating to the exposure of rats to cats or to cats' odor or to the confinement of the animals into cages or the disruption of the sleep pattern or the maternal separation. And we have found a number of interesting results with regards to oxidative cascade. And all these results can be resumed into that the trauma can cause depletion of the antioxidants, level function, and increase in oxidative stress byproducts. Going through the animal studies and reaching to human studies and the early life stress studies, we can find studies on health controls and on personality disorder cohorts and on psychotic cohorts, all these combined with the element of childhood maltreatment. And we can see in the picture a number of effects which can be summarized that the childhood maltreatment can correlate later in adulthood, later in life, with increased oxidation activity. And going to severe life stress experiments and studies, we have found that a study on survivors from an earthquake or studies on survivors from an air catastrophe witnessing or cohorts diagnosed with PTSD all have shown a number of interesting results with regards to oxidative cascade. And these are very indicative of the high complexity of the oxidative mechanisms. And of course, we have results from war-related trauma from American Gulf veterans showing increased peroxide nitrite, and from Croatian veterans diagnosed with PTSD with decreased levels of dismutase and glutathione peroxidase. And last but not least, we have gene studies on oxidative stress, again with water veterans cohorts, which have shown a number of genes which are candidates for regulating oxidative stress and have shown highly association with PTSD. These are the retinoic acid orphan receptor alpha, the glutathione S transferase, the thioredoxin reductase, and the alloxygenase. But apart from these genes, there are a couple of other genes, benevolent ones with known neurotrophic features like the brain-derived neurotrophic factor, as well as the clotho gene, which is an age suppressor gene, which have shown that some SNPs can relate to vulnerability, whereas other genotypes can relate to resilience to PTSD. And physiologically speaking, there is a perfect balance between the pro-oxidative molecules and the antioxidants, whereby we have low concentrations of calcium glutamate and nitric oxide, as well as basal function of these neurotrophic elements like N-acetyl aspartate, BDNF, and GABA receptors. Whereas, pathologically speaking, when stress, trauma, injury, and inflammation happens, then we have these at risk of this oxidative equilibrium with the decrease of antioxidants and an increase of free radicals, whereby we have a significant increase of these toxic components, as well as, in contrast, significant decrease of these neurotrophic factors. And leaving the first question, what is the relationship between oxidative stress and trauma? We go through another question about what is the relationship between oxidative stress, inflammation in the context of trauma. And for us to comprehend this, we need to delve into basic sciences and see how the pathogens are functioning with damage and pathogen-associated patterns invading within the cell and being recognized by the pattern recognition receptors, and afterwards the pro-inflammatory cytokines are being secreted by activating the innate immune system. And here we can see the innate immune system and the adaptive immune system. And here we can see the pro-inflammatory arms of both innate and adaptive immune system and the anti-inflammatory arms. And as long as inflammation is getting dispersed, subsequently, the stress response is getting orchestrated with the activation of sympathoadrenomedullary pathway, as well as the activation of hypothalamic pituitary adrenal axis with the final secretion of glucocorticoids, offering the well-known anti-inflammatory action. And when reviewing the whole literature, we can find evidence about an inflammation-driven oxidative stress stemming from trauma, which activates microglia, toll-like receptors, as well as upregulates sympathetic nervous system and causes dysfunction of the glucocorticoids. And with intermediate stages, we can find the secretion of pro-inflammatory cytokines, leading finally to the activation of oxidative cascades. In contrast, we have found evidence for the opposite direction, this being the oxidative stress-driven inflammation, which originates from oxidative components, which can consequently activate elements from inflammation like G and K, nuclear factor kappa beta, glucocorticoid receptors, and glutamate, which finally end to the pro-inflammatory cytokine secretion. But the most probable pathway can be this hypothesis of the synchronous activation of both inflammation and oxidative cascades stemming from trauma through toll-like receptor activation, leading back to finally to simultaneous activation of pro-inflammatory cytokines as well as reactive oxidative species. Finally, we need to be minded of the limitations of these research field of the differential trauma type, recall bias, sample size, comorbidity, and genetic and epigenetic variability. And going through the time, we can see Hippocrates, who gave place to Galen and subsequently to Freud, and now we're going to the modern times with the contemporary psychiatrist who is called to handle new research fields of psychoneuroendocrinology, psychoimmunology, oxidative stress. We're pretty much confident in the modern psychiatrist's capacity that can put the psychiatry in middle of organic medical specialties and psychological sciences, so that psychiatry will finally take the place it deserves. Thank you very much.